Okay, it's my pleasure and honor to introduce tonight's speaker, Dr. Doug Pace. Doug is assistant professor in the biology department at California State University, Long Beach. He grew up in rural New Jersey and developed a fascination for nature. He completed a Bachelor of Science in Marine Biology at Long Island University, Southampton College, and completed his PhD at the University of Southern California, where he studied how environmental conditions, such as food availability, temperature, and hydrostatic pressure, affect protein metabolism in developing marine invertebrates. During his studies, he conducted research in Antarctica and at the hydrothermal vents of the East Pacific Rise. At CSULB, he supervises a lab that studies the developmental physiology of marine invertebrates. The title of tonight's talk is Making Sense of the Arms Race, Phenotypic Plasticity During Sand Dollar Larval Development. Thank you, and help me welcome Dr. Doug Pace.
Yeah, I think so. Everyone hear me okay? All right. Well, thank you so much, Julie, for the introduction, and thank you for the invitation. This is really an honor to be here and get to talk to you guys about my research. So let's get going here. So making sense of the arms race, we're going to talk about larvae, and we're going to talk about phenotypic plasticity. And so here we go. So presentation overview, I just want to actually tell you who I am, a very brief overview, and then actually give you a little, hopefully a convincing argument why you should be paying attention to anything I'm talking about tonight. Because I do appreciate your spending your Friday evening, listening to me talk about my research. And I want to talk about why larvae matter, why plasticity is a phenomena that is ubiquitous and very much worth our scientific attention. And then I want to talk about some of the research that's going on in my lab right now, and really the product of fantastic students that I get to teach and work with and mentor at Cal State University, Long Beach, both master's students and undergraduate students. And through that process, I will bring you through 3 research questions, as shown here, talking about plasticity in marine invertebrate larvae, and then talking about the questions that we are pursuing. And I think we're getting pretty far in answering these questions. And then I will conclude with some future directions for the lab. Okay, so there we go. Who am I? So actually, I didn't realize the bio that Julie read pretty much answers a lot of those questions already. But I grew up in northern New Jersey. And I want to brace yourself because this person you see here did not always look like this person you see here. So there I am. Can you tell which one's me? This guy over here. So yeah, I think I'm like all of 14 years old at that point. I definitely was very much in my, I don't know how to say this gracefully, my dirt bag headbanger phase. But it was a great time for me to grow up. I really spent most of my waking hours in the forest. My friends and I, we would build log cabins. Just what the heck? Let's build a log cabin. And of course, we had a pet raccoon because, everybody has a pet raccoon, right? It's just what you do when you grow up. So that was great. And then, I got a little older and realized I should probably do something useful. So I went to Long Island University at Southampton College, and that is shown here. And that's where I got my degree in marine science. And that's really where I became, interested in a lot of the things that drive my current research today in a general sense, not in a very specific sense. And I'm sad to say that this wonderful college is now bankrupt, or went bankrupt a long time ago. So I don't really have an alma mater. But I will say that I feel like it because they're always hustling me down for money, right? I don't know what's going on, but they want me to donate to a school that doesn't exist anymore. So after that, I then went and worked for a year, pretty close to where I went to school at Brookhaven National Laboratory. And this was my first real exposure to research. And I worked under the direction of Paul Kemp. And we were trying to understand, he was, and I was really a minion there, trying to understand, can bacteria in the ocean actually suck up carbon from the atmosphere and store it away? Is this a reliable way to take care of all the carbon dioxide that, as I'm sure you guys know, we are producing. And this was called the Joint Global Ocean Flux Program. And then after, that was really important for me because I learned that I loved research, but I also learned I didn't like bacteria, right? And it's really important to understand, what you like to do and what you don't like to do. So with that information, I then went on to the University of Southern California, where I got my PhD studying larvae, and this is really where I became interested in things that were very specifically about larval biology. And so the title of my dissertation is Feeding and Protein Metabolism in Developing Marine Invertebrates. This is an amazing opportunity, a time in my life. I studied many, many different kinds of larval forms you see here. And as Julie mentioned, I had the opportunity to dive in the famous submersible, the Alvin, several times. And we would collect these hydrothermal vent worms and bring them back to shipboard, and then we would try to grow their embryos. And we were the first lab to ever successfully do this. I can tell you that I was personally there helping take this SEM photo, the only SEM photo that exists of a baby Riftia, a baby tube worm. So that was really, really exciting. And then after that, I stayed on so that I could go to another really cool place, which was Antarctica. So this was my postdoctoral experience, where I studied protein metabolism. And these guys down here, these are Antarctic echinoderms, and they do very, very well underwater in Antarctica. And really our goal there collectively was to understand how life can exist at such cold temperatures. And there I am in such cold temperatures. Then for some reason I decided I was going to make a hard left turn. And I decided that I would take a chance and study something completely unrelated to everything I had done up to that point. So I went to the University of Georgia, and specifically the Center for Tropical and Emerging Global Diseases, and I decided I would try to be, or maybe imitate, a molecular parasitologist. This is pretty wild, right? So I walked into a lab and I knew nothing about molecular techniques, genetic modification of organisms, and certainly nothing about human intracellular parasites. And it worked out pretty well. I learned a lot. I basically broadened my scientific knowledge base by a tremendous amount. And although I don't study this parasite anymore, it has taught me how to be a much better biologist and ask much better questions and look for the techniques by which to ask them and answer them. And if you're wondering, this parasite here, this tiny little thing you see in these little bundles, that's living inside a human cell. 30% of this room has this parasite, right? So I'm just going to walk and not talk about that anymore, and I'll let you guys think about that. But I will just say we've learned to get along with this parasite very well. So you're all okay if you are holding this parasite. Okay, so then in 2013. I guess I fully became a grown-up and I got a real job. And Cal State University Long Beach hired me. And so I've been there ever since. And this is the beautiful CSULB campus, where I'm running what I call the Ecophysiology Larval Lab. And these are just a smattering of some of the students I've had really the privilege and the honor to mentor through the years. The students affectionately call themselves Team Larvae. And I get behind that all the way. We even have merchandise. I can sell you a shirt if you want one. But it is a real bonding experience. And it's really great to get to work with students and teach them some of the tools of how to ask questions, how to answer questions, how to get data, how to interpret data, and learn about the world around us in a systematic way. So what is it that we study then as Team Larvae? We study larvae. So the thing I really want to take a moment is hopefully convince you guys that larvae are important. Otherwise, everything I say after this really is meaningless, right? So what are larvae and how are they important? They are, in a biological sense, an early part of a life stage. They are an immature form of an animal. And this is the stage that exists prior to some dramatic metamorphic event. After that metamorphic event, the larva then turns into a juvenile, which will then become the adult. The larval stage typically is pre-reproductive. So there is no reproduction going on in these larval stages. So this really is, if I can get the direction right here, this really is what we would call an indirect form of development, okay? And so what that means is we have direct developers. Direct developers, that's us, right? And mammals, right? We come from an egg, the egg is fertilized, and that egg becomes an embryo, and then it goes through gradual change and takes on the aspect of an adult, right? And this is what we are very familiar with. This is our personal experience with development. So things with a larval stage are indirect developers. They have their egg, and that egg, after fertilization, will become this embryo. The embryo will then go through gradual change to take on this larval stage, right? And then this larval stage will grow and develop and go through a metamorphic event and become the juvenile, which looks a lot like the adult, and there'll be more gradual change. And so we have these two distinct forms of development, direct development and indirect development. And the question really becomes, why pursue an indirect form of development? Why have a larval stage at all? Generally, it is to accomplish something that will not be accomplished in the adult stage, right? And so there are many reasons for this, but two of the big ones are simply get big, right? So you're in this larval stage and your job is to eat as much as you can and grow and get bigger and bigger and bigger so that when you do turn into this juvenile after your metamorphosis, you're big and you either can predate on things very easily or things cannot easily predate on you because of your size, right? We can think of the hungry caterpillar, right? The children's story, and the caterpillar just ate and ate and ate and ate. So this is a really important function of the larval stage that helps the emerging adult to survive. The other one is also just to get away from your mom and dad, right? And I think as teenagers, we can all understand that, right? So this is really about some organisms benefit massively by having a lot of gene flow. They have a lot of gene resources and variation in their gene pool, and this gives them a form of protection so that if there's a local catastrophic event, there's a huge amount of genetic variation in that population by which some will certainly die, but many will still move on, and the species as a whole will continue. This is a very, very important part of what the larvae can do in certain organisms, especially the organisms I'm talking about tonight. So because of this large amount of variation in function of larvae, they pursue a very large amount of variation in their morphology, right? And so this is what I call the bizarre world of larval forms. All of these things don't look like the adults that they're going to turn into, and they also don't look like each other, right? And so that's because they're pursuing different goals in terms of this larval stage. And so evolution has fine-tuned them or directed them in ways such that their morphology helps them do whatever it is that their specific task is in terms of being a larval form. So the other reason why larvae are important, and this really, I think, is a very important reason. It's so important I even put a pie chart there, right? So you know you can believe me because there's a pie chart there. But there's about 35 phyla of animals out there, right? And of those 35, at least 30 of them have at least one species that has a larval stage. So when we're talking about life on Earth, it's really important to understand how pervasive the larval stage is. And really, when it comes to complex animals, metazoans as we call them, larvae are the rule. They are not the exception. And I know it's easy to think that because we think, oh, a larva, a caterpillar turns into a butterfly. That is a good example, but there are many, many more examples. And so ecologically, evolutionarily, developmentally, this larval stage is an incredibly important part of life on Earth. Now, the larval stages I study are from benthic invertebrates. And I think a lot of you are familiar with photos like this. You're members of the aquarium, right? And we have some beautiful benthic communities off the coast here in California, and tropical reefs and Antarctica again. And so we understand that benthic adults, they don't move around so much. right? Their level of motility is relatively minor compared to the vastness of the ocean. And so the larval stage here becomes incredibly important because this is an opportunity for larvae to be planktonic, meaning they float in the water, and they can disperse great distances from where they came from. And so this is a major, major role that marine invertebrate larvae play. The larvae that I'm studying and is the focus of this talk is none other than this beautiful Pacific sand dollar community here, Dendraster excentricus, and I assume many of you are familiar with this guy. And so this is a burrowing irregular sea urchin, mostly found in these sandy benthic habitats. They play a lot of important ecosystem roles: bioturbation, oxygenation, and of course they are a very important food source for lots of other things that are critical to our environment. And this is the larval stage here, okay? And this is, just so you know, this is not the scale. This larva is probably no bigger than the width of your hair, just so you can understand the size of what we're talking about. Now, when we look at these communities, we know that Dendraster is located really all up and down the west coast of the United States. And so we understand that they will reproduce, and these larvae are very, very small things floating in the water, right? They have some motion, but they can only control their really local environment. For the most part, this is a particle that is being picked up by the ocean currents and swept away to certain locations. So their dispersal capacity is great. These larvae can live for about 2 to 6 weeks. They can travel great distances. That could be good. It could be bad too, right? So there's lots of chances that these larvae are going to end up in locations where life is not going to work out for them. But it also is a vital source of resupply of adult communities, right? So as adult communities age and members die, larval recruitment plays a huge role in resupplying those communities and in also making new communities. Now, one of the things, though, is there is a real distinct problem with this plan for this particular larva. These larvae are feeding larvae, meaning that they cannot grow and they cannot develop unless they get food, right? And this brings me to a horrible realization I made in grad school. The oceans are filled with the worst parents. Terrible, terrible parents. What do? Well, this is what they do. I mean, this is in a lab, but this is a pretty accurate sort of depiction of what goes on in the wild. This is a male white sea urchin spawning sperm. This is a female spawning eggs. And that's what they do in the natural environment. It's a broadcast spawn. They all just go out there and they float in the current. And if an egg and a sperm are lucky enough, they will meet. And they'll be like, hey, I like you, you like me, right? And then that egg will become fertilized and they will develop. And this is all happening while they're carried on a much vaster current, right? It's something they have very little control over. And if they're lucky and everything works out, that embryo will gradually turn into the larval form. And the larval form, and the energy, I should say, provided by the egg, that is the only parental investment. That's all they get. And that is just enough energy to make 2 things, really: a stomach and a mouth. That's it, kid. You're on your own, right? And so that's what these larvae are up against. They're being pushed and carried by currents they have little control over, and they're entering environments that may have food, may not have food. It's really hard to know. And so what do we study in our lab at Cal State Long Beach, Team Larvae, is we look at the developmental physiology of these life forms. And our real question is an ecophysiological question. We want to understand what are the tools these larvae have so that they can roll with these punches. Oh, there's no food today. What will I do to survive? Okay, now there's a lot of food. What should I do so that I take maximum advantage of these environmental changes? And so we look at this through the lens of physiological energetics and functional morphology. So here is a depiction of the life stage of the sand dollar, and I will say they spend much more time looking like this, but I find them much more interesting when they look like this. And so basically you have broadcast spawning, fertilization in the water column, and if things are good, this is a really kind of standard developmental trajectory. And what you may notice is there's something growing over here, right, on the side of its stomach. That is what we call the adult rudiment. That is the part that gets to move on to the next stage and become the adult. The rest of the larva is cast aside. Maybe some of it is reabsorbed, but most of it is cast aside. And so this rudiment then turns into this cute little fuzzball here, and that is a juvenile sand dollar that will now gradually change and take on what you see there is the adult sand dollar. And so what we are interested in is how do the larvae contend with environmental changes they have no control over and make it all the way to this point so that they can get through metamorphosis. So here's an up close and personal look of a larva. And I want to show this because it shows you the basic structure. This is what we call a pluteus larva. And I hope you can see it, but there's a ciliary band on this larva, and it circumscribes the entire larva all the way, just wraps all the way around it. That ciliary band is responsible for allowing the larvae to move on a very small spatial scale, and it creates a feeding current. And I think you can see these little particles here. And so it's creating a feeding current because these little particles are algal cells. This is what it eats. And so it's trying to create this current so that these algal cells will go to its mouth, and it can eat, and it can grow and develop, and it can be happy. Now, the project we're pursuing is phenotypic plasticity, right? And so I want to give a brief definition and background of what is phenotypic plasticity. Phenotypic plasticity is what we would call, as scientists, a gene by environment interaction. And so if you think of some silly movie in the 80s, like I think it was Danny DeVito and Arnold Schwarzenegger were identical twins, right? And then they were separated at birth, and they went through very different childhoods. And therefore, they ended up one looking like Danny DeVito and one like Arnold Schwarzenegger. That, oddly enough, as ridiculous as that is an example of phenotypic plasticity. What it means is from that same genetic background, going into different environments creates a different response. And that's the response of the genotype interacting with the environment. This is a very, very critical thing to be studying, especially now with the amount of change going on in our environment. There's rapid change going on, especially in coastal environments. And these organisms need to have some toolkit in order to roll with these punches. Phenotypic plasticity is one of those tools we imagine. So it represents a crucial diversity reservoir acting as a first response mechanism. Okay, and so this may allow an organism to survive immediate changes in its environment by changing its phenotype to match the environment. So a really good example are these two guys. This is textbook stuff we would show in a classroom, right? These are called water fleas, Daphnia, and they exhibit very extreme morphological plasticity. Both of these are genetically identical, or similar, we'll say, but they don't look the same, right? And so you can see this guy over here has grown up in the presence of a predator. And it is said, I'm going to make some defenses, these spines, to protect myself from predation. This guy over here did not grow up in the presence of a predator and said, why would I waste my time and energy making these spines? So this is a classic case of phenotypic plasticity. Now, what does it look like in the larvae I study? Well, what we have is, because these larvae encounter very, very different food environments, they are able to sense changes in how much food is around them. And when they sense these changes, they change their morphology accordingly. So if a larva enters a high food environment, it grows very short arms, because how fast the larva can eat is dictated by how long the arm is, how long that ciliary band is, how strong is that current being made to capture the algae so it can eat it. So a high-fed larvae will not spend a lot of energy making long arms because it doesn't need to. A low-fed larvae will take a very different direction and actually increase the length of its arms to try to offset or compensate the low food environment it is in. Okay, and so basically I'm going to make a longer ciliary band, and I'm going to create more water current, feeding current, so that I can try to eat more of the low amount of food that is available. Is that a question? Yeah. Okay, if you remember, I'll be up here, and I'm really excited to hear what your question is. Okay. All right, so this is seen as adaptive, right? And the reason for that is, as I said, low fed, you make your arms longer to increase your feeding ability. High fed, you don't waste the energy doing that. Okay, and so what we really want to know, and this is, by the way, very quickly, this is how a scientist looks at it, right? So we have what we call midline body length here and the arm length here, and we try to measure them and standardize them. Because here's the thing, if I give you more food, you're going to grow bigger, right? Because you just have more food. And what we're looking at is a response that goes beyond that. So we take our data and we standardize it to the average size by doing this midline body length. And so what we look at in a graph like this is for the same midline body length, here's where the high fed is for their post-oral arms, and here's where the low fed is. And so what we're seeing is a distinct, different growth strategy. For the same size of my total larval size, I'm going to make much longer arms. And that's what plasticity looks like to somebody who studies it, okay? Now, the idea, though, is that this has been studied for 30 or 40 years. And what I really want to do with my physiology training is understand what else is going on here. Because clearly, changes in form are going to be related to changes in function. It's like our textbooks, anatomy and physiology, right? When anatomy is different, physiology will change and vice versa. Yes. Oh my goodness. I feel like I planted you right there to ask that question. So you and the rest of the room, he wants to know if you switch the food, will they switch what they look like? Oh, if you go into the middle? Well, you know what? That is a great question. And we only look at the two ends, the low and the high. But we should do that. And actually, I have a student over here, and that will be her project. So that is a great idea, and we are working on it. Wow, all right. So there has to be more, right? And here's a ridiculous example, right? I like to teach using ridiculous examples. Here are some giraffe, and by the way, thank you, ChatGPT. They are eating leaves from a tree, right? And so let's just say something ridiculous happens, and the trees suddenly become much taller, and the giraffes somehow have this phenotypic plasticity where they're like, no problem, I'm just going to grow a longer neck, right? Okay, so we're looking at that, and we're like, geez, I think there's a subtle difference here. Their necks just got a lot longer, right? But do you think that's the end of the story? Thank you. Thank you so much. Yes. Yeah, so if we made the trees shorter, well, you'd have to ask them. I don't know. This is all from the world of ridiculous examples, so it's hard for me to know. I am, and I just wanted to focus on the trees. So if I may. So the idea here is you look at this and you be like, yeah, their necks are longer, right? But surely there has to be other things going on to compensate or to support this kind of radical change. Anatomical changes in muscle structure, innervation, physiology. I mean, my goodness. Think about the heart down here trying to pump blood all the way up there, right? So there would have to be some major changes here. And so I looked at this as a field that really wanted to just measure arm length of larvae, and I felt like we really should be pushing this much farther, and as I would say, getting under the hood, looking at the engine, and understanding what else is changing so that we can appreciate the integrated response here, right? And so here it is. Here we are with our 2-day-old larvae. He senses high food. He turns into that eventually. He or she or it senses low food and pursues this morphological strategy rather than this one. These are very, very different strategies. And so the first question I wanted to talk about tonight was how different are these strategies? So yes, the arms are getting longer, but is that really where this morphological change begins and ends, right? And so a graduate student here, Wendell Lee, she then decided that she would try to understand this by looking at three-dimensional morphology of these larvae. And I should say, actually, why don't we, you know what, you're going to like this. Here, this is a three-dimensional, 3D printout of a larva. And I really just, if I can I trust you guys? You can pass it around and go, ooh, ah, thank you. I think he'll need to see it first. And so she wanted to understand what's going on with the volume of these larvae and the surface area of these larvae, right? So the first thing she did was look at their biomass. How much protein do they have? Now, I just want to get you acquainted to this graph. On the graph here, we have the x-axis. This is just how old they are. Are they 4 days old, 8 days old, 12 days old? So we're looking at this as they age, and we're measuring how much do they weigh? What is their biomass, right? So this is really not a shocker. If we feed them a lot of food, they weigh more in blue than the ones we feed an order of magnitude less food, right? This stands to reason, right? If I give you more food, you're going to get bigger. But what she wanted to do then is looking at this, what happens to other aspects of their morphology, such as their volume, and more importantly, their surface area. And so this is really, really interesting because now taking these same larvae here and graphing how their surface area is changing over time, what you see is this massive change in protein doesn't show up. By extending their arms, they are greatly expanding their surface area, okay? Now, this is interesting to a physiologist because so much of what an organism does physiologically, it scales to their surface area. What is their exposure to the world around them? And so this set up further questions that she pursued to understand what are the consequences of this massive expansion of surface area. So here we know, hey, longer arms, cool. We can feed better. We can try to offset this low feeding environment by having longer arms and feeding more. But now we see longer arms is also giving you greater surface area. What are the consequences of that? And what we see is a mixed bag. We see that increased surface area has stuff that's good about it, and it has things that maybe are not so good about it. Yes? That is a very, very good observation. I believe what he said was with increased surface area, they're a bigger target, right? And yeah, they could be food very easily. Yes, that is excellent. And so we're not studying predation, but maybe we will in the future. But what we want to look at are what are the physiological consequences here, okay? And so here is the larva. Again, this is, thank you, ChatGPT. It's floating in the ocean, and it's sharing this space with a heterogeneous environment. Lots and lots of things are in the water. The food it wants to eat is floating in the water, but there's a ton of dissolved stuff in there. A lot of that stuff that's dissolved is actually pretty good for the larva, dissolved sugars and amino acids. And these are things that the larvae has molecular transporters for, and it can grab these things from seawater and bring it in and put it to good use. And so this is great, right? Amino acids and sugar, right? You look on the back of your favorite, cookie, and you can see how much protein and how much fat and how much sugar is in it, right? The larvae likes the same stuff, okay? And so when it gets these things from the seawater, it can use that for energy production, and it can use that for growth. And so if you increase your surface area, you increase your ability to grab these goodies from the seawater. This is an adaptive response. This is a good thing. But the problem with seawater is it's seawater, right? And seawater is loaded with salts, right? And salts are very, very disruptive to the metabolism and to the development of an organism. And this high gradient of salts is always trying to get into the larva and get into the cells. And this is true for us too. And so the larva has to spend a tremendous amount of energy kicking the salt out of its cells and putting it back into seawater. And as you increase your surface area, you increase that stress too, right? So there's a good side to this, and then there's a very problematic side of having to spend a lot of energy to kick the salt out because now you've increased the surface area by which the salt can get in. So what you have here is a trade-off, and evolution loves a trade-off, right? And so basically, you've got aspects that are good, but it's not all for free. There's always something that has to be paid for that response. So the response here is good because you have access to dissolved organic nutrients, but you've got to work hard to keep all the salt out. The hypothesis that came from this work is that this is probably something that works to the larva's benefit beginning in early development. The good part of this outweighs the bad part. But as the larva develops and the arms get longer and longer, we feel that this switches, that at some point, the increased surface area exposure is actually bad because of the salt regulation. The ion regulation is what we would call it. And so this puts forward a hypothesis that we can then test, and it also says there's going to be a cap to this strategy. These arms just can't grow ridiculously longer and longer and longer, right? And so this sets into motion the second question I want to talk to you about today, which is what is going on physiologically with this life form? We see these arm changes. We see these changes in surface area, right? So what's going on if we crack open the hood and look at the engine and understand things like physiological efficiencies? And so these are some of the students that helped address these questions. And this was a major ask. This was a very, very tall order. And so basically, they became larval dietitians, all right? And thank you again, ChatGPT. I could make these wonderful, ridiculous images. So here's a larva on a treadmill. Here's a larva getting weighed, some very, very interested doctors, and so, but this is really what we were doing, all right? Now I'll put it into boring scientific terms. And so here is the kind of trend we're trying to capture and understand. And what do? We measure how much they eat. The blue is high-fed, the orange is low-fed. Again, no mystery there. If I feed you more, you eat more. And then we look at, well, how much energy are you using? This is the ridiculous idea of larvae on a treadmill, right? And again, if you're bigger and you're growing more, you're going to have a higher metabolic rate. And then we look at growth. And again, if I'm feeding you more, you're going to grow faster. The blue is high-fed and the orange is low-fed. And again, all these x-axis are the same. It's looking at how old are the larvae, how are they changing as they grow and develop. The key to this, though, is we're not learning anything here, really, right? If I give you more food, these are the changes we would expect. The key is to look at these changes and standardize them and begin to look at what we would call physiological efficiencies. And so what we can do with this data is make what we call an energy budget. And so here, we're modeling, this is how much food you took in as you developed. And then in the orange is how much food you used and invested into growth. And then in blue, how much energy you use, your metabolic rate, as you develop. And what we do there is we take these ratios. We try to make standard comparisons. And so what we do here is this is assimilation efficiency. How much energy did you use, plus how much energy did you put to growth, and standardize that to how much did you eat. Okay, so this is a percentage, right? And then we also did growth efficiency. And here it's a simpler version of this where we just looked at how much did you grow compared to how much did you eat, right? So if I ate a pound of food and then I grew a pound, my growth efficiency would be 100%. And that would be troubling, right? That would be really, really troubling, right? So, but what we want to understand is these low-fed and high-fed, are they exhibiting physiological differences when we look at these growth efficiencies or digestive efficiencies? And what we see is, yes, they do exhibit differences, right? And so here is the low-fed. Assimilation efficiency is quite high, and then it decreases quite readily throughout development, and the growth efficiency is very similar. Not quite as high, but it is high, and it decreases. The high fed, shown in blue, a completely divergent pattern. And we see the high fed actually starts at lower efficiencies and increases as they develop. So this is very interesting because this, again, looks exactly like the trade-off that was our original hypothesis. That early on, this expanse in surface area and morphology is going to work to the larva's advantage as a low-fed, but as it gets older, it's not going to work nearly as well. And that is exactly what we're seeing here, is it's all really great for the low-fed larvae, and then at some point, its efficiencies drop below the high-fed. This is telling us that this is a good strategy early in development, but it's a strategy based on diminishing returns because the cost involved in sustaining the strategy eventually becomes too great. So early on, things are great, benefits of increased feeding and dissolved nutrients, and then the penalty of the increased energy you have to spend to keep the salt at bay overshadows any benefit you were getting. Okay, and so this is really exciting because the hypothesis that we generated then really was supported. Okay, it's a hypothesis, we can't prove it, but we can find data sets that at the very least support it. And so this really, we think, is an outcome of this increased surface area exposure, something unavoidable for the larva if it wants to grow longer arms. Okay, so the final thing we did is then ask, after all of this journey, is this response actually adaptive? And the reason we ask that is because not everything an animal does is adaptive. And it's important that we take a moment and really try to understand things can respond to the environment, but not always in the best way. Okay? And so the two questions we're looking at here is this plasticity response, much like this young gentleman asked earlier, can it happen throughout development? If I take a low-fed larva and it's halfway through its development and then I switch it to high food conditions, is this response so beneficial that it will then change its morphology and pursue this high-fed, short-arm phenotype? The other question is just how much energy do they use throughout their larval development, and do any of these changes matter in terms of how much energy is required for them to finish their development? And so we understand the ocean is heterogeneous, and food changes quite a bit through space and through time, and what we want to know is if this is really adaptive, these larvae will track those changes. Low-fed for low food environment will have long arms, and a high-fed larvae will have short arms. Can they do this, pull off this trick throughout their larval development? And so what we did here is look at this change and take these low-fed larvae and switch them to high food conditions at 10, 20, and 30 days of age. This was a really surprising and a very, very cool result because it was pretty obvious what was happening. We're looking at the length of the arms, and as soon as we take this low-fed larva and put it into a high food condition, it immediately is like, get these long arms off me. I want nothing to do with these long arms. They've been a liability all along, and now that I don't need them, I'm going to shrink them and get them out of my way. And this really was, it's one of those happy results that you don't need to be a brain surgeon to interpret. You can see this pretty clearly. So the next question then was, and that really backs up the idea of this is a trade-off, and therefore they don't want the long arms unless they absolutely need them. So in closing, the last thing we did was just look at how much energy is required for a larva to go from that egg all the way to metamorphosis. Okay, because that's really where the rubber meets the road. How much energy does it take? How many gallons of gas does it take to drive from the beginning to the end? And so what you see here is this result. Whether you were high-fed all the time, switched at 10 days, or switched at 20 days, the amount of energy it took was the same, even though it was twice as long for these guys to develop the switch at 20 days. This is a very adaptive response. This means that because of the low-fed response, they were able to save energy and hold on and wait until better times arrived. This poor guy, though, what this is really showing you is if you're low-fed all the time pursuing that strategy, it's not going to be good for you. you're going to have to spend an awful lot of energy as you wait longer and longer so that you get big enough so that you can metamorphose. So what we see here then is really a more nuanced explanation of how this is adaptive. It's adaptive, but it doesn't solve a problem. What it does is allow the larvae a potential plan to withstand these times of low food, low abundance, and hang on so that when high food does come, they can take advantage of it and get rid of those long arms. So take-home message, the arms race is real, but it is so much more than just looking at how long or how short an arm is. And the reason why this is important, again, is that given the amount of change that is occurring in our coastal habitats, we need to have some understanding of how to do appropriate conservation biology, right? And now that we know that larvae are ubiquitous, most of these life forms have a larval stage, and they're so important for population recruitment and keeping populations there, we want to understand the adaptive mechanisms by which these larvae are going to be able to respond to these changes. And so phenotypic plasticity is most certainly adaptive, but it's complicated and it's a gamble, right? Like a lot of things. You make a choice on the best available information and you hope things are going to go your way. When a low-fed larvae puts on these long arms, and they look beautiful, right? But when they do that, they are putting themselves in danger of lots of other things, but they're doing this because they're hoping high food comes along. So this is an organismal physiological response. It's not just how long your arms are. There's so much more going on underneath that. Biochemical, molecular, and genetic mechanisms. And so very quickly, because of that, the future for our lab is really bright. We have so many ideas we want to pursue. This is a collaboration with my good friend and collaborator, Dr. Siavash Ahar, and we're looking at the hydrodynamics of larvae. And what we're doing here is tracking fluorescent particles, and it looks like starry night, right? And you can see these beautiful vortexes they're making, and we're trying to understand when morphology changes, how does the intimate relationship of that larva's environment changing with it? Other projects we have, we're looking at molecular mechanisms. We understand that these changes, although they're physiological and morphological, they're being supported by differences in how genes are being expressed, right? And so this is a volcano chart looking at genes that are downregulated and genes that are upregulated. The bad news there is there's an awful lot of genes that we might need to look at. But that's job security, right? We're also looking at biochemical energetic reserves that fuel morphological changes, and this is a current graduate student in the audience, actually, Troy Stern, is looking at that. And we're looking at predictive modeling of plasticity responses, because what does it matter if I can tell you what a larvae did in my lab? It doesn't matter much. What we need to do is take everything we're learning and plug it into a dynamic energy budget model. And this is research being done by Lorelei Cunningham, also in the audience. And so this gives us the ability to make predictions about, as the environment changes, how do we expect the larvae to behave, and how might that have downstream consequences on population stability? And then finally, we're looking at how heritable is this trait? And this is working with my departmental collaborators, Dr. Bruno Pernay and Darren Johnson, looking at, is this a trait that can be inherited? And if so, that means natural selection can work on it, because some larvae might have a better plasticity response than other larvae. All right, we got there, I think. So in closing, I just want to express my gratitude to you guys listening to me. And then really, this has been a great experience working at Cal State Long Beach. I have got to interact and work and mentor and maybe give some good advice to so many students and send them on their way to bigger things. And in the process, learn a little bit about marine invertebrate larvae. I also want to thank the funding sources. Support your local funding sources like Southern California Academy of Sciences. I'm a board member, so I have to make a plug for that. National Science Foundation, NIH, and COAST has been amazing as well. And then in closing, thank you for your attention. I understand that, the opening ceremony is being broadcast, so I'll bring it to you with a slightly larval flavor. ChatGPT, it really does some strange stuff. And so, of course, Peace always. All right. Thank you so much for your attention, and I'd love to take some questions.
Thank you, Doug. So we can take some questions from the audience. Thank you, Zach, for making the mics hot. The mics are hot. Okay, so just remember, you guys, they can hear.
Yeah. Question off topic, but do these larvae, well, if they change into a juvenile and they're a couple 100 miles off the coast, they would sink down and not do very well. If they are near the coast and they change into a juvenile, they would sink down and become sand dogs. Is there a column effect in the metamorphosis of the larva?
A column effect?
In other words, can they sense how deep the water is?
So that's a great question. And this is really a lot of other people's research, but I certainly do read up on it. So one of the $1 million questions for a lot of these studies are, what is the cue? When does a larva understand that, first of all, it's developed enough so that it can successfully get through metamorphosis? And then when is the right time for it to do that? And so I don't think they're necessarily dialed into hydrostatic pressure, like where they are on the water column. But what I think they're looking for is a sign that this is the right place. And so for a lot of these larvae, that sign is they kind of smell the water, right? And they understand there's people like me there. They get the signal of conspecifics, right? They're like, some of my brothers and sisters or aunts and uncles, right? They're around here somewhere. I smell them. And that is enough to make them basically initiate metamorphosis. And when they do that, the effect of losing the larval mass and only having that adult rudiment, that makes them negatively buoyant. And so they actually settle down quite naturally. Now, results will vary, right? So, you have to have a couple of things happen all at the same time. The timing of development when they're able to metamorphose has to match with where the currents are taking them. And then they have to find that community where they want to settle down. And so the reality is, as I showed you in that one image, there's a female echinoderm can spawn millions of eggs. And what one colleague of mine at USC used to call it is the sweepstakes approach. Basically, 1 in a million. So maybe 1 out of every million that actually come from that clutch of eggs actually makes it to become an adult, right? So it is a very complicated process, and we're trying to learn more about those molecular cues that actually allow a larva to understand when is the right time. But even if they get the cue, they still have to make sure they're actually developmentally ready to make the change. Yeah. So a lot of good things has to happen. Does that help?
A little bit, yes.
Yeah. A little bit? Oh, okay. Yes.
So they are real-time adapting to their environment. Is that like a DNA thing, or is it instinct? And if it's instinct, can you define what that is?
So that's a great question. We think it is a molecular mechanism. Basically, they're sensing a chemical, and this chemical we think is actually dopamine. It's actually a neurological molecule that we use in our everyday life, in our brains and whatnot. And for them, dopamine signals that there's algae. And so when they're getting algae, they're getting dopamine, or they're activating a dopamine pathway, and that actually tells them keep the arm short. And then as the dopamine goes away, it actually releases that prohibition on arm growth, and the arms grow longer. That is our current understanding of that response. And how fast that response can happen is still being looked at.
So this is all the other animals, the other percentage are larval stages, the mollusks and polychaetes, they're all acting like this?
So yes, and thank you for asking that. This plasticity response is seen across the board in marine invertebrate larvae. Not necessarily all larvae, but it's seen equally, kind of distributed around different kinds of larvae. So in an oyster larvae, it doesn't grow longer arms because it doesn't have arms, but it grows a longer velum or a wider velum so that it still makes a longer ciliary band by which to gather more food. Yeah, so this really does seem like, in a general sense, a ubiquitous strategy. Yeah.
If you put a long-armed larvae into a high food environment, how long does it take until these arms shrink? Are we talking hours, minutes, days, months?
Well, we thought we were asking that question appropriately. Now it doesn't want to go. Okay. Can I operate this from here?
Yeah.
Okay, okay, cool. All right. Oh, I'm trying to get out of the presentation. Sorry.
You want to get out of the presentation?
Well, it's easier for me to move around, but that's okay. Now, how do I get rid of this? I've really done it now. More.
High floating.
There we go. Okay, let's just do it this way.
You can go up and down arrow.
Okay, so that, and I probably went over this a little too quickly, but that actually, I think, is addressing your question. So this is the time frame where we took the low fed and put it in the high food,
Right.
and that's how quickly it dropped. So that was taken 2 days later. So that response, we know, takes at least 2 days, or at most 2 days. It could be even faster. We just didn't measure it fast enough. So this is a pretty immediate response. And so they are tracking these changes in their environment. And that makes sense because that would keep them, it would make this response an adaptive one. Yeah. Yeah.
Do they drop it or they reabsorb it?
Well, we don't know. We think it actually, what we think is that the amount of energy spent just to keep it that long is tremendous, right? And so what it is there's turnover. That tissue is going away very quickly, but they are taking great pains to keep replacing it, right? And you can't see that. It looks like it's just, there. But what's going on underneath all that is degradation of the tissue and replacement of the tissue. And the moment they sense better times, they stop replacing the tissue that is degrading. And then what's amazing is these are skeletal arms, calcium carbonate, that just dissolves, right? Because these are very, very thin calcium carbonate arms, and that's how they do that. But we have to check that to be sure. So, yeah. He was just stretching. Okay. Yeah.
So this is like about food availability for like are there any other factors that might
Really?
like warmer water, chemical pollution exposure? Would that also affect like water?
That's a really, really good question. Yeah. So what I showed you was how these organisms can change in response to food levels changing. But there's lots of things changing in their environment, not just food levels. And the short answer to that is we don't know, right? But we are looking at it. In fact, we've got Ethan over here, who is looking at this response when you increase temperature, right? Because if environments are changing and there's an average increase in temperature, well, how will that affect what looks like a pretty good adaptive strategy as it gets warmer and warmer? Right, and so stay tuned for that. We don't have an answer quite yet for that. Yeah. You ready? Okay.
If the gravity changes, if it has long arms, wouldn't the gravity change? So the gravity is longer because the gravity every object has some gravity.
Yeah. Okay. So
the long arms, the little gravity can hold some stuff next to it. But with the tiny arms, he only has a less radius because
I like what you're thinking there, right? So as they get longer arms, they might get pulled down faster. No? Okay.
sticks on them because they don't fall. So the longer radius of the gravity, but not longer, but like it should fall in a longer.
So the thing about that is those kind of things might happen if you're in the air, but when you're in water, especially when you're that size, it's hard for us to imagine. But when you're that size and you're in water, you are basically in motor oil, right? Because, and this is what we call the Reynolds number, right? And this gets into some fluid dynamic physics. But basically, the smaller you get, the more impactful the viscosity of the environment you are in. So we get in water and we're like, yeah, we're swimming, it's cool. They are so small that the way they interact with water, it is like they're in motor oil. So they're in a large way, they're kind of being suspended by it, and it's not nearly as easy to move through as you might think.
You mentioned earlier the several different potential stimuli that might cause a larva to go into metamorphosis. If you're getting metamorphosis in the lab, they're not getting any of those stimuli. They're going solely on food intake, I would guess. Is that true, or are there stimuli that you can use in the lab that can trip them into metamorphosis?
Yes, so you can get them so developed that they just run out of options, right? And they will spontaneously metamorphose, right? Because they can't hold off forever, right? It's kind of like, all right, I'm ready, I'm going to hold my breath, and I'm going to wait for the perfect place. But the perfect place never shows up. And so we can do that in the lab easily enough. We can induce them to metamorphose. These guys are pretty straightforward. If you just take sand that had adults in it and you put that in the culture, that smells like adults. And they're like, well, this must be a safe place because other adults are living here, and that can get them to metamorphose as well. But that's not true for all species. Some are we just don't understand what their cue is. We know they have a cue, but it's very difficult to find.
So Doug, let's take a couple questions from home. Okay.
Okay, yes.
Okay, so Carl, who's former staff, we miss you, Carl, and he loves echinoderms. He asks, is there any indication that this plasticity responds to other factors such as salinity and water density?
Carl, there is no indication because no one's looked at it. But we actually, so I have another graduate student who's looking at I don't know where to look. Carl, are you there? Yeah, so I have another graduate student who's actually looking at salinity responses, or how the food-induced plasticity response changes when they're in different salinities, right? So we're still looking at the food-induced response, but acknowledging that salinity is a changing variable as well, and how do those two interact?
Okay, another one from home, Julie. Do the arms keep their length, i.e. do the arms continue to be long as they begin metamorphosis?
Great question. And actually, they do not. And that is in this graph right here. This blue is constant high fed, and you can see what's happening, and you can see it down here as well. As they begin in the latest stages of their development, as they make the rudiment, the arms actually do begin to get smaller. Yes.
Carl's got one more.
Okay.
He says, fascinating work. Do you think that this can impact the larvae's defense cloning response?
He wants to talk about cloning. Okay. So that opens up a whole nother realm of larval biology. But what we have learned is echinoderm larvae, and I kind of simplified things, and I said it was pre-reproductive. Some of these larvae can actually clone, right? And it's pretty mind-blowing because what actually happens is a little bleb can come off their arm, be released into the water, and actually turn into this larva, a smaller version of the larva, right? We do not really have a firm grip on cloning and whether or not it's actually sort of a lab-based artifact. We're doing something that is causing a response that really has very little to do with the natural ecology of the organism, or is this actually something that happens and it's a way that larvae are able to reproduce in this larval stage? And because of that, I don't know how phenotypic plasticity plays into it. Yeah. So future research for the next 80 years. Yeah.
How about maybe 2 more questions from the audience?
Okay. Yes, yeah.
Have you guys looked at any of the maternal input? So have you standardized like the yolk that starts all of this and seeing if that has any implication to the size and how they change? Yeah.
So, I am typing madly. This study here. So this is a really cool study, and what we've done is we've actually used the hot dog roller you see in your local 7-Eleven, right? This is pretty amazing. We basically are growing single larvae at a time in a little 20 mil chamber, and it fits into the hot dog roller. So it just rolls and keeps them well mixed. And so what we're able to do there is grow hundreds of individual larvae. And so we do hundreds of different crosses, this male with this female, this male with this female. And then we can model this using math. And that's really where this guy, Dr. Johnson, comes in. And we can actually look at how much of the plasticity response is heritable. But then we can also look at how much of the plasticity response is coming from the mom. Because the major input that all larvae get, the thing that really they inherit from their parents, is the egg, and that's from the mom. And the mom gives the egg, the larvae, the biochemical substrates, the energy, but it also gives it lots of messages, molecular messages about how to develop. And so we think that that does play a major role. Our current estimate in terms of how much it interacts with the plasticity response is about 13%. But it is definitely important, yeah. Yes?
But what if, because if we could clone, then what if you take the cut a piece of the arm off and then take one of the parts, a tiny bit of the part off on purpose, would it clone?
Would the arm grow a full larva?
No, because if you have a arm, put a little bit of, and then some of it goes, and can it turn into a larva? Because you said if sometimes the stage go off,
Right.
but if you cut them off, will it turn into one?
Usually not.
You know?
Yeah, the cloning has to happen all by itself, and it happens through a way that we don't understand. So if I just go in and cut the arm off, it'll just fall and look sad and go to the bottom.
Yeah, but it'll evaporate.
What will happen to it?
These are the paper, so would it just evaporate, turn into water?
We should talk in the we should talk in the gift shop, right? Is that right?
Go to the gift shop. Yeah, let's end it there. So please join me in thanking Dr. Pace for a fantastic lecture. On behalf of all the staff, we would like to give you this little gift.
Wow.
Yeah, thank you again. What a wonderful lecture.